Hi, everybody. It's a pleasure to welcome Dr. Fauci to the ACTG and other CAB meetings of NIAID CABs to update us today on coronavirus 19. We have been trying to get this scheduled, and we're so happy that you can join us today, Dr. Fauci. I understand you have some prepared slides for us, and then we have some questions that the community has already asked, and we'll go through those.
Sounds good. Thank you very much, Carl. Let me just get right to it. First of all, it's a pleasure to be with all of you today. We're living in strange times that we're going to have to do everything virtually, but I think we can do it in a way that hopefully will be helpful and interesting to you. Having to do things virtually, we're living in somewhat of a little bit of a surrealistic existence because I still don't know what date it is today. I don't know if it's June or March or February because it's not advancing. Because it seems like yesterday or 10 years ago that the Chinese made that announcement that they'd identified a new strain in Wuhan, which we all remember. Like I said, it seems like so long ago. We moved quickly in that we screened travelers at airport. If you look at the dates on this Washington Post papers from the 17th to the 20th to the 21st, and yet we still had travel-related cases 2 days later on January the 21st, first detected in the United States. And then we all know what happened. We had a global explosion of cases that have now reached extraordinary proportions, with over 7 million cases and 400,000 deaths in the United States. Literally, as of 2 days ago, we had over 2 million cases and over 112,000 deaths. Truly historic in every sense of the word pandemic. That is something that I've often said to the press, and it's true. Really the worst nightmare of a highly transmissible virus that's brand new, that has a significant degree of morbidity and mortality. So we at NIH and at NIAID, even though it's a multi-institute approach, we're going to focus predominantly, but not exclusively, on what NIAID has done. We have a strategic plan that really follows what we do with every outbreak, dating way back to outbreaks that we've done back in the very early 80s when we first developed our strategic plan for HIV/AIDS. And it's based on 4 fundamental principles. The first, a fundamental understanding of the viral biology, the pathogenesis, the development of animal models. A couple of examples as shown on this slide. The basic research always informs where we go with the ultimate interventions. We need to understand viral biology, life cycle, transmissibility, natural history and surveillance, particularly in a highly transmissible disease, is extraordinarily important, as are animal models. And something we do that's very important is we serve as the source of research reagents and readily make them available to the entire national and even international research community. We also develop diagnostics and assays. One of the, I believe, potentially very important approaches is the considerable amount of resources, $500 million, that have been devoted to a program called the Rapid Acceleration of Diagnostics, or RADX. And what it is a national call for innovative technologies which we judge in a rapid selection process, which some have referred to as a Shark Tank-like process. We quickly go into phase 1, phase 2, and hopefully, by elimination of those that don't work, we will get to a couple of winners, hopefully several, by the end of the summer and the early fall of 2020. We also characterize and test therapeutics. This slide is multiple bullets talking about a number of potential approaches, from direct antivirals like remdesivir, other broad spectrum, hyperimmune globulin, repurposed drugs, immune-based therapies, such as convalescent plasma, monoclonal antibodies, hyperimmune globulin, and others. This is a map of the national and international network of trial sites that was used in the adaptive randomized controlled trial for COVID-19, which is an adaptive trial that we will continue to add on to. The one that we referred to that was the first randomized placebo-controlled trial was one that we recently published in the New England Journal. It was in remdesivir. It was over 1,000 individuals. The results were significant but modest, with a 32% faster time to recovery in hospitalized patients with documented pulmonary disease. We use that as a starting point. Now another trial, which is comparing remdesivir alone with remdesivir plus baricitinib for COVID-19. That is an ongoing study that is about 50% enrolled. We also put together a guidelines panel because people on the outside who are treating every day people with COVID-19 wanted to be able to have a central clearinghouse where the information, which is coming out every single day in peer-reviewed and unpeer-reviewed journals, can be synthesized in a readily available document that's a living document that can change and be updated on a very regular basis. And finally, development of a safe and effective vaccine. These are some of the vaccine platform technologies, from the genetic immunizations of the DNA and RNA, and the companies that you see are on the right-hand side of the slide, viral vectors, anything from adenovirus to VSV, recombinant proteins. At least a couple of companies are doing that. BARDA, or the Biomedical Advanced Research and Development Authority, is supporting a number of these vaccine candidates. The ones that BARDA has committed to supporting are shown here. One of them is a Moderna product, which is an mRNA platform that was developed at the Vaccine Research Center at NIAID. It is rapidly advanced into clinical trial. It is a typical mRNA in which the gene for the spike protein is inserted in the mRNA, injected into the muscle, which then expresses the protein to induce an immune response. The timing on this was extraordinary. The Chinese put the database up with the sequence on the 10th of January. We had a meeting on the 11th. We started the trial on the 14th. 62 days later, we were in phase 1. I said we started the trial, but we started to develop the vaccine, not the trial. The trial started 62 days later. The phase 1 has been completed, and that was the fastest we've ever gone into a phase 1 trial, shown of the differences in the various diseases that we've approached, from well over a year to now a matter of just a couple of months. The results are promising. They will be published soon. Good neutralizing antibodies at reasonably good titers in the phase 1 study of 45 individuals of 3 separate doses of the mRNA. Larry Corey, John Mascola, Francis Collins, and I published in Science a couple of weeks ago our strategic approach to the COVID-19 vaccine research and development to go at a pace that is fast but with no compromise at all of the scientific integrity or of the safety of the individuals, and to scale up at a rapid way using harmonized protocols that we will be able to compare results, common DSMBs, common laboratories determinations, to be able to have cross checking and cross comparison of the results of one trial to another. The approach that has now been referred to as Operation Warp Speed, as I've always said, I would not necessarily have picked that name because some people misinterpret it as meaning we're rushing on something and possibly compromising, and we're not. This is a national program to accelerate the development and the manufacturing. It is headed by Monsef Salawi, who's someone we've had experience with over the years when he was at GlaxoSmithKline, and we are moving along very well. Francis Collins has initiated a program called ACTIVE, or Accelerating COVID-19 Therapeutic Interventions and Vaccines, in order to get outside individuals, public-private partnership, to prioritize, accelerate, and do the clinical evaluation of both therapeutics and vaccines and diagnostics. And it's moving along extraordinarily well. We've put together what's called a NIAID coronavirus prevention network, where we've essentially taken the three networks that we previously supported for both HIV, influenza, and other infectious diseases, and put them together into a consortium so that we will continue to prioritize projects in the HIV prevention as well as the non-COVID vaccine work. So we're using things we previously built for other purposes and utilizing them now in a very important purpose. This is one of the examples of the prevention network. We have multiple platforms going on in our harmonized efficacy trials in collaboration and cooperation with, as I mentioned, the Common Data and Safety Monitoring Board and the statistical groups, so that you have a common look at the data. Finally, COVID-19 in persons with HIV, obviously something, given our history, that we pay considerable attention to. It is unknown if people living with HIV have an increased incidence of infection. There's a large study that's testing people with HIV for SARS coronavirus, and they found no difference in incidence, interestingly, that's related to either the viral load, viral CD4 T cell count, or whatever antiretroviral drug regimen they're on. Certainly, that persons with HIV with comorbidities are more vulnerable to the complications of COVID-19, which is very similar to the general population that may or may not have comorbidities. The complications of COVID-19 occur at a younger age in general in persons with HIV compared to the general population, and one wonders whether that has something to do with the premature aging phenomenon in some HIV persons with HIV. And the same minority groups that have a disproportionate incidence of HIV have a disproportionate incidence of severe coronavirus disease. So we're going to continue to look at this very carefully and very closely. Finally, you all know that I'm a member of the White House Coronavirus Task Force. It's a group that meets regularly and goes over any of a number of the issues that are both scientific as well as a variety of other public health approaches. This is just a picture of what I do almost every day, namely down at the White House. This is the Situation Room in which we're briefing President Trump. And this is just an example of where we are right now, which is in the process of trying to open up America again. And you know, this is going to be something that we have to do with great care because we want to make sure we're able to respond to those inevitable spikes that we will see as people try to get back to some degree of normality. I'll stop there and Carl, if you want to moderate the questions, I'd be more than happy to answer what we can in the remaining time. Okay,
There we go. I am now unmuted. Thank you. It's a pleasure to see all of you virtually. Can you still hear me, Dr. Fauci?
I can hear you fine. Thanks, Carl.
So we have a number of questions that have already come in. First, how are you? How are you holding up? And how is everybody in your family?
Well, I sound like crap. I can tell you that much from my voice, because I give so many briefings and so many podcasts and so many hearings and things that I've never been able to get the irritation of my voice back. But I'm feeling reasonably well. As I've told you, Carl, many times, I talk to you every day. I'm running on fumes, but the fumes are pretty thick. So I think that we're going to be okay. Everybody in my family's fine, getting a little bit harassed by the radical element in our society, but we can handle that. So not to worry about that.
That's incredible. So here's a really kind of an off-the-wall question. You've been interviewed by several celebrities recently. Which were some of the more memorable interviews you've done with some of these really interesting characters?
Well, that's a great question. We've had some really good interviews from groups that I never would interact with. Rap groups, for example, who have a following that I didn't even know existed, like Lil Wayne. And then I did Steph Curry, which got another different group. I got onto the Instagram of the chef Jose Andres. That to me was just fantastic. And I find that the audiences that you get from them are so different than the CNN audience and the network audiences. So I think I'm going to continue to do those kind of outreach. You get more into the community by doing that.
That's thanks so much. So you and I were around when the HIV epidemic began. What lessons have we learned from HIV that can be applied to the COVID-19 epidemic, both in general and in terms of the social stigma related to the disease?
Well, I think you hit upon the last one is really important. I think the disparities to me are so similar. We know, and we live with HIV every day now. We haven't given up on it at all. We're still very much involved in it. And the disparities are still there. If you look at the population in general, demographically, African Americans make up 12-13% of the population. They have the definitely disparate burden of HIV. If you look at the COVID-19, both in the incidence of infection because of the social determinants of health, which put African Americans and other minorities, including Latinx and Native Americans, in a position that they are more susceptible to infection because they're in jobs that put them into contact as opposed to being able to lock down and get in front of a computer. And when they do get infected, they have such a higher incidence of the comorbidities that are responsible for the high degree of morbidity and mortality. That's a striking similarity. The other similarity is that in the beginning of HIV, when we were trying to work out pathogenesis and clinical manifestations, they were protean. We were getting surprised at things we were seeing. Same thing now. I'm continually marveling at the spectrum of disease of COVID-19, from no symptoms at all to some mild symptoms that you don't even notice, to symptoms that bother you a little, to symptoms that keep you home, to symptoms that put you in the hospital, to symptoms that put you on a ventilator, to symptoms that kill you. It's very rare that you have a single infection caused by a single pathogen that can do everything from nothing to kill you. So that to me is one of the big differences. And also the explosive nature of it is something that continues I marvel at how efficiently it spreads from human to human.
That is pretty amazing. So one of the interesting things that you and I have talked about is we are in the process. This disease is less than 7 months old, and we have not really had the time to truly stand up a community engagement program. One suggestion that has come in is we have the COVID-19 treatment guidelines. Do you see this as an opportunity where community could engage and participate in the guidelines committee and help us move forward as well as the kinds of activities you and I have talked about, is bringing community into our clinical trials as we go forward.
Carl, I think we should immediately, we're already doing it, but I think we should absolutely go for it to do exactly what we did back in the day when we brought the community into every aspect of what we do. The guidelines, the protocols, everything. Because if we don't engage community the way we've engaged it with HIV, we're not going to do as good a job as we could do. And we're all in on that.
I think that's so true. And thank you for your answer. In conversations with people who live with HIV, doubts have been doubts around having HIV research networks on COVID-19 keep coming up. They believe that the work toward HIV cure and vaccine has entirely stopped, and that federal money is being diverted from HIV research to COVID research. Will you help me please address these concerns?
Yeah. Okay. So I can promise everybody on this call that we are not taking our foot off the accelerator when it comes to HIV. I mean, HIV is such an important infection, and HIV/AIDS is such an important disease domestically and internationally, that our efforts to end HIV in the United States is still going full blown. We're still involving our networks in that to a great degree. So please don't think that anything is being pulled back. It's not. The second part of your question, I think, is important. We are certainly not taking HIV research money and diverting it to SARS coronavirus 2. As a matter of fact, there's a lot of SARS coronavirus 2 that is coming into that we really don't have any need at all to poach money from any other of the groups. We've been very fortunate in that the Congress and the administration have been very generous in giving us the money that we need to carry out our research program on coronavirus. HIV is not suffering either with effort, with attention, or with resources.
Thank you for that. At the same time, though, we have directed the HVTN, the ACTG, and the PTN to get engaged heavily in our coronavirus research. So I think it's important for people to understand that we feel they have the bandwidth to do both. You want to expand on that?
Yeah. So we are so proud over the years that we have been building up, perfecting, and fine-tuning our network, which everybody knows is the best, the biggest, the most unprecedented clinical trial networks ever for any disease. The fact that we built it in a way where we incorporated the best and the brightest of clinical trialists, of people interested, starting off with HIV, who've shown they can easily and readily adapt themselves to contributing to the research in another very important disease, at the same time as manifesting that they have the bandwidth to continue the effort of what they're doing in the primary disease that we're involved with. I think that's a testimony to the flexibility of the entire network process, as well as to the individual investigators who show they can do more than one thing at the same time.
I would like to echo that too, and remind everybody that we were able to complete the HPTN 083 study that was reported out. Hopefully you didn't miss that news. And the major vaccine trials, the 705, is continuing on. So we remain committed. We are essentially out of time. Do you have time for one more question?
Sure, Carl, why don't you go ahead? Why don't we take one or two more?
One or two more. Okay, I'm trying to pick the most important ones. So here's an interesting one. We know that HIV virus lives in reservoirs, which makes it hard to find a vaccine. Has the coronavirus been found in reservoirs such as lymph nodes or the brain after a person has received or infected with the virus? And are we concerned about persistence with the coronavirus?
You know, Carl, the answer is we're still learning. We have to be humble to know that we don't know everything. But what we've seen so far does not at all point to the kind of profound impact of a reservoir that we see in HIV that is not what we're seeing. We're seeing that even when people recover, they have positivity by PCR, but that is almost certainly not live replication competent virus. It's nucleotides that are dead, that are there, and will ultimately get cleared. There's no evidence whatsoever that a person who gets infected, recovers, clears the virus, has a reservoir that we're not detecting.
Thank you. I think one last question that I think is on everybody's mind, and I noticed that you have been talking about this recently, is while we move forward and develop a vaccine that will need to be distributed, how are we going to assure that the people who need treatments and vaccines can obtain them and afford them? And how are we going to move this forward collectively as a global operation to make sure vaccines and therapies are available to all?
Well, as you know, there's nothing that we can do to force companies regarding pricing, but we have an agreement that as we give the government assistance, which we're giving a lot of money to the companies to de-risk their getting involved in a risky business of trying to develop a vaccine, an understanding that when a vaccine is developed, that we will make sure that that is made available at a reasonable cost. And for those countries that can't afford it at all, that the rich countries will make the donations that are necessary to get everyone and anyone who needs this type of intervention to have it. This is something that we are committed to. And when we talk to the companies, we get the firm impression that they are also very committed to that.
Well, thank you for your time today. Do you have any last comments to the community? And I would like us to just thank the community and encourage them to get out and get engaged with us to help bring a community voice to coronavirus disease. That's something that we desperately need, and HIV can lead the way. Would you agree?
Yeah, I totally agree, Carl. And I want to take the opportunity to first thank the community reps and the community individuals for everything they've done for so many years that we've been working together. But to underscore what Carl just said, that we are a better enterprise when the community is involved with us. We know that. We've proven that over and over again with other diseases, particularly HIV. I have no doubt that if the community is all in with us in COVID-19, we will do a much, much better job of doing the kind of service which we've been trained to do and have been doing. So thanks for what you've done, and thank you all for what I hope you will do. Take care. Take care of yourself. Stay safe.
Bye, everybody.